Our final session of the afternoon is our other viral infections in relation to COVID, and the moderator is Dr. Roberta DiBiase. Dr. DiBiase, as we know from this morning's introduction, is a tenured professor of pediatrics and microbiology, immunology, and tropical medicine at the George Washington University School of Medicine and Health Sciences. Dr. DiBiase is the chief of the Division of Pediatric Infectious Disease and co-director of the Congenital Zika program at Children's National. So Dr. DiBiase, the session is all yours to lead.
Thank you so much. We're going to close out the day here with looking at COVID in relation to other viral infections. And to me, this is really great because if you think back on this symposium, I think last year or the year before, I've lost track of the years, we were so focused on what is this thing? How is it affecting our patients? Anything we could just learn about, having our head directly in the moment. And now it's really clear when we hear these talks today that we're moving beyond that acute kind of crisis response and really able to pull our heads out and look and see how this fits into the context of everything else that we're going to have to deal with over the next few years. And any of you that are clinicians know that we're now seeing all of these other viruses roar back. We're going to see newly emerged infections. So we're really excited to have these next three speakers who are basic scientists, someone involved directly in emergency response, and then a really well-known journalist and author to put this in context. So I think you're going to enjoy this last session. So our first speaker is Dr. Lisa Hensley. She is the Associate Director for Science at NIH's Integrated Research Facility in Frederick. Prior to this, she also held positions as the Director of Regulatory Science for the Medical Countermeasures Initiative in the Office of the Commissioner at the FDA, and also as the Chief of Viral Therapeutics at USAMRIID. She had her PhD at the University of North Carolina, Chapel Hill, her master's in immunology and infectious disease from the Bloomberg School of Public Health at Hopkins. And she spent the last 20 years developing and characterizing animal models for high consequence and emerging pathogens, including Ebola, Marburg, SARS, MERS, orthopox, and other viruses. She's used these models to develop and evaluate and help advance candidate countermeasures, whether that be therapeutics, post-exposure prophylaxis, vaccines, diagnostics, and beyond the confines of her own maximum containment lab here domestically. She's worked in West and Central Africa as well as other parts of the world to help them develop their research laboratories to respond to these outbreaks, because of course it's in these areas that many of these infections may emerge or proliferate because of the lack of access to resources. She's considered one of the world's leading subject matter experts on emerging diseases and high consequence pathogens, and we're just thrilled that she's here today. And she's going to compare SARS-CoV-2 to other coronaviruses. What is it that makes SARS-CoV-2 different? So the floor is yours, Dr. Hensley.
Thank you. It's a pleasure to be here, and I want to thank the organizers for the invitation and the opportunity to speak today. And also, of course, I also want to give a nice shout out to our previous speaker. I am so impressed, high school student. That was a great presentation, and to come up here and give it or to do it remotely, kudos to you and to your mentors. Just a great job. So let me just also dive in to say my disclosures. I am a U.S. government employee. I just recently transitioned about a week ago over to the USDA to set up their new zoonotic and emerging disease research unit. And of course, I have the typical disclosures of any government employee. So today we're going to be talking about coronaviruses, and looks like our screen's lagging a bit. I might need some help, guys.
If you click on the slide and then try to advance, that should work.
Okay. Okay, great. Sorry, guys. I think I missed that in the chat. So today we're going to be talking about coronaviruses. All of us have become coronavirus experts. We've learned quite a bit about this virus over the last few years. So I'm not going to spend too much time on this, other to say that coronaviruses are a family of single-stranded positive sense RNA virus. They're very large viruses for an RNA virus, but they also have very similar genome organizations and structures. And coronaviruses are not new. They've been around for a number of years, but I think for the most part, we thought of them in the veterinary field, or we thought of them in the agricultural field. So when you think about coronaviruses, I think the first one that was recognized, and there's a debate on this, might have been feline infectious peritonitis virus. It wasn't until the 1960s that we recognized this as a human pathogen. So what's important to keep in mind? When I think of coronaviruses and I'm explaining coronaviruses to people, Coronaviruses affect a wide variety of animals, including wildlife, domestic, and also agriculture. Fun fact for the day, there's even a coronavirus found in whales. All coronaviruses are zoonotic, meaning they originally evolved in animals and jumped over into humans or still are in animals, and that the host range and tissue tropism are largely defined at the point of entry. So what do I mean by this? What I mean is that the ability of the virus to infect is really driven at that point of that virus attachment to the cell and the ability to get into this or to get into the cell. How do we know this? Well, you can take the RNA from these viruses and you can introduce it into most cells and you can establish a productive infection. So the point is the bottleneck for cross-species transfer and for development of disease really is at this very early stages. Now, there's going to be some other factors, and I'll talk about those later on, that will impact how well the virus can replicate and whether or not it establishes a productive infection. So this next slide here just lists the receptors for human coronaviruses as well as the entry pathways. And as I alluded to in my last slide, how well the virus is able to use receptors as it crosses species, or how similar the receptor is across different species, can be a key determinant of how efficient viral cross-species transfers are. You will sometimes hear this as receptor homolog scanning. Back in the 20s, Ralph Barrick and his lab, when we were studying cross-species transfer, they had first started using this term. And when you think about coronaviruses, and you think about COVID-19 specifically, we think it likely came from bats. There might have been an intermediate host somewhere in there, and it has caused this massive pandemic. But as we've learned, it also infects a number of other species, and we've seen it becoming endemic in white-tailed deer. We've seen cases in domestic animals. I think there was a horse a few weeks ago, exotic animals. But what this tells us is as this virus has jumped species, and as it jumped species, it acquired the capability for a broad host range. Now, the other important point about the receptors and the ability of the virus to use the receptors is that the receptor distribution may drive or does drive the tissue tropism and may drive the pathogenesis or the disease that you see. And I think the simplest example for those of us who don't spend a lot of time working with viral entry or thinking about these things is when you think about transgenic mice. And this is an example that's going to be very familiar to all of you, which is with the COVID transgenic animals. When we overexpress the ACE2, we saw that it changed the disease course. So by overexpression and having it in the brain, we saw more of a neurologic disease course develop. And I'm so sorry, guys. It seems like that my son's dog was very quiet right until I had to give this talk, and all of a sudden has decided to join our conference. So I'm going to apologize in advance. So I mentioned also at the beginning genome organization, and I said that these viruses share a very similar organization. So why am I wasting a slide on this and having you look at this? Well, one of the things that is different about many of the viruses is the accessory proteins. So you see this very common structure, but if you look at the slide graphically, it does show you that the viruses aren't all the same. And there's a number of these other proteins, many of which we don't know their entire purpose. Some have been well characterized, but these different proteins likely play a pivotal role or a critical role in how the virus causes disease, how it's able to evade the immune system, and how it's able to maintain the infection. So this was a nice slide. I pulled it off of the Scientific American Journal, or this case website. And this is just a timeline. And I wanted to put this slide up here to talk a little bit about human coronaviruses. So to date, there have been 7 human coronaviruses that have been detected. There's NL63, 229E, HKU1, OC43. And then there have also been SARS, MERS, and SARS coronavirus 2, or COVID-19 for simplicity for this lecture. So when you think about those first 4, these are often referred to as low pathogenicity or seasonal coronaviruses. We heard a little bit from Dr. Seti about how people can be reinfected with these. And these are viruses that we're going to see over and over again, and they typically cause mild disease. The last 3 are ones that we consider to be of pandemic potential and to be of higher pathogenicity. So just briefly, a little bit of similarities with the human coronaviruses. So when you look at the seasonal ones, all have very similar incubation periods. Again, they're typically considered to cause very mild disease, typically respiratory, but you will also see GI symptoms. Approximately 10 to 30% of the common colds are attributed to these seasonal coronaviruses. And again, we routinely see that these viruses that you'll see them over and over again, that you can be reinfected with them. All of the coronaviruses I mentioned are likely zoonoses and have jump species, likely so an intermediate host. And you can see the picture over there, some of the intermediate hosts that have been portrayed. And importantly, there's likely other coronaviruses that we haven't even yet recognized. And I'm going to spend just a minute or two on some of the seasonal coronaviruses. So NL63 likely originated from bats. Predicted hosts are bats and palm civets. It uses the ACE2 for infectious entry. That's a typical the receptor for it. It's a seasonal coronavirus. And why I wanted to pop this slide up here, I'm not going to go through it in depth, but why I thought this one was important to highlight was that when people looked for this virus or identified this virus, for the most part, they thought it caused mild disease. But in the early cases that it was identified, 8 of the 11 patients that tested for this virus, or they identified this virus, also had Kawasaki's disease. So here's an example of a seasonal coronavirus that typically causes very mild disease. Now, there are exceptions, again, people who are immunocompromised, other underlying conditions, but it's an example, depending upon who gets that virus, there may be a very serious disease course, and you may also have neurologic outcomes as well. When you look at 2 of our other coronaviruses that are up here, some of the seasonal ones, again, very mild disease. And I wanted to point these 2 out, again, looking at their evolutionary identity, but also indicating that they're relatively new for us, right? We didn't identify HKU1 until January of 2005. My guess is it's been widely circulating. So these viruses, we haven't known about them for too long when you think about how long we've been studying viruses, yet these are viruses that are all around the world that we see on a routine basis. The last one that I'm going to spend just a minute on is OC43. So this is the first human coronavirus that was identified. And I mentioned that they weren't identified until 1967. And there's this large gap. So why was this large gap? Well, again, back in the early 60s or the late 60s, early 70s, we didn't think too much about coronaviruses for human disease. It seemed to cause only a mild cold, mild illness. It was thought to be something really for the vet field or the ag field. And of course, all of that changed when SARS happened. So when SARS occurred in November of 2002, it was really for many of the scientists who are focused on human pathogens, viruses, that to really think of coronaviruses. And I remember I was doing my postdoc at USAMRIID at the time, and the scientific director, when it was identified what SARS was, said, and he had a very colorful language, but fill in the blank, fill in the blank, fill in the blank. It's a something coronavirus. And where the hell am I going to find a coronavirologist? What are there, like 5 of them? And he wasn't too far off. For the most part, the coronavirus field had been slowly dying. I had done my PhD on coronaviruses and actually had left studying coronaviruses up until SARS, mainly because the advice that there really wasn't going to be much money in this. But post-SARS, we realized that coronaviruses could cause significant disease. It spurred on looking for other viruses, which is why we now have several more other coronaviruses identified. But for whatever reason, SARS was easily or readily comparably easy to control. So SARS was able to be controlled. It did spread to a number of countries. There was over 8,000 cases with 774 deaths in 28 different countries. But it went away and things were quiet for about another 10 years. And then MERS happened. So MERS evolved or was first identified in the Middle East. To date, there have been over 2,400, 2,500 cases and 858 fatalities. But this is a virus that doesn't seem to spread well. For whatever reason, transmission is very infrequent. It seems to have come from camels. It seems to have come over in multiple cross-species jumps or spillovers, and we're still continuing to see cases, likely from new crossover events. And of course, the last one is COVID-19, SARS-CoV-2. So again, we're probably way too familiar with this, so I'm going to use this as an opportunity to segue into my next slide, which is really what I want to talk about today, which is why do we see these differences between coronaviruses? Why are all coronaviruses not created equal? Why do you get a mild disease with one? Or why does one virus rapidly fizzle out or burn itself out, yet another one turns into a global pandemic? And what can we learn from this? And what can this tell us from the future? So there's a number of different theories here. There's a number of different potential options and ways that may drive the differences between the coronaviruses. And I'm going to talk a little bit about some of these. Okay, so my first one, whenever I talk about why viruses, why you might see differences and why one virus becomes a pandemic or grows and the other one burns itself out. And the first one is transmissibility. So how well does the virus replicate? How is it spread? What is the route of transmission? How easy is it to prevent that route of transmission? How well does the virus grow? If it grows to much higher titers and there's 10 to the 8th or 10 to the 9th in the blood or in the saliva, as compared to 10 to the 4th, 10 to the 5th, how does that impact the ability of that virus to spread? How easy is it to detect someone and to isolate someone who has the disease? And then how stable is the agent in the environment? Is there the risk of fomites? Will it be around for some time? Now, when you look at SARS and you look at MERS and you look at COVID-19, about 22 to 40% of the SARS cases and 13 to 21% of the MERS transmissions occurred between family members. So it's actually a fairly small number there when you start thinking about it. And when you look at the number of healthcare workers or infections that occurred in the hospital, it was actually much higher. So what is this telling us? It's telling us that if 62 to 70 to 80% of our cases may be occurring in the hospital setting or after clinical disease has already onset or has been after clinical disease has already developed, it's telling us that that virus is going to be easier to control. Whereas COVID-19, we're seeing transmission before the onset of disease. So that makes it much more difficult to control and to isolate. Viral fitness. So several studies have linked the possible higher transmission of COVID-19 compared to SARS or MERS to how well the virus is able to use the receptor. And for SARS-CoV-2, it's about 10 to 20 times better than SARS-CoV. So when you think about that virus jumping species and that idea of using the same protein but across different species, there's going to be changes in that protein. So how well can that virus still use that protein? How well can the virus get into the cell? Can it use the host proteases to help it get into the cell? These are things that are going to affect it. How is the receptor distributed? Is it similar? Is it different? Is it in different tissues? The next thing that I think about is interactions with the host. Does the virus trigger the innate system? Can it use the cellular machinery? Does that protease that it needs, does it work? The transport gene, is it able to utilize it? Keep in mind, viruses are replicating in the cell. They need that host cell machinery. Can it evade detection by the immune system? And can it spread or go to the next person before it kills the host? So probably the thing that comes to mind first is escaping the immune system. So coronaviruses are notorious for their ability to control or to suppress the interferon system. And there have been a number of papers on this. And I think some of this, it might have been surprising because many people have looked at interferon as an antiviral, even for coronaviruses. I even did that myself years ago. So what does that tell us? One is that this virus is uniquely adapted to controlling the interferon response, and how well it prevents either the production of interferon or the response to interferon may in fact drive whether or not it is able to cause disease. And so one of the things that about SARS and MERS is the replication strategy actually allows them to help avoid some of that interferon response. But it's not just interferon. So there's also data coming out looking at how the virus controls apoptosis, how it may drive cellular death or how it may prevent cellular death. And that ability to control these responses will be critical in driving whether or not it causes severe disease. My favorite is dysregulation of the immune system, having spent years on viral hemorrhagic fevers. This idea that when a person gets infected and the virus goes into some of these target cells, is it able to go unnoticed by the cell, or does it set off a cascade of events? Does it set off a cytokine storm? And I think we've heard a lot about cytokine storms with COVID-19, with SARS-CoV, with many other viruses. But what happens? Is it going into the macrophages? What cell types is it infecting? And what is being produced? Is it making reactive oxygen species? Are those triggering other downstream events? Is the reactive oxygen species causing inflammation? Is it causing damage to the lung tissues? How is this resulting in the pathogenicity? And then, of course, once you have this system turned on and you have this dysregulation that's occurring, the inflammation will trigger the coagulation. It will create disruption, and this is going to continue to cause problems in the host. So next slide. So when I think of this, and somebody asked me, why is this virus causing disease and why is this one not? Why is this virus going into a pandemic, but another one is not? To me, it's multifactorial. There's not a great answer. Certainly, we can point to different proteins that are in COVID-19 that are maybe not in other ones or receptor usages. But I found this figure, and it was actually put together much more eloquently than a summary that I had. So I've adapted to this, and I apologize to the authors. The reference is cut off. But when I think about it, it's a combination. There is the host factors, the genetic background. There's the ability of the virus to get in. How transmissible is it? There's the ability of the virus to adapt once it gets in. We know coronaviruses recombine. We also know that they're capable of mutation. Being a larger RNA virus, they might be a little bit more plastic than other ones. We also know that the viruses have this unique and very complex relationship. So if it can use the host cell machinery, if it can control immune responses, if it can control inflammation, or maybe it wants to trigger inflammation, these are the things that are going to drive. And that delicate relationship will drive whether or not it becomes a severe infection, a mild infection, or grows into a pandemic. So trying to wrap this up, and a little food for thought here at the end of it, my question is, are we missing more coronaviruses or are there more to come? And I will upfront say there likely are more to come. But I threw this out as an example, mainly because it's been in the literature recently and a lot of people have started talking about it. But if you look, the earliest coronaviruses were identified in the 1960s. But if you look, you'll see that there are actually a lot, tip of the iceberg, over 400 different viruses in bats, in China alone, wide diversity also in Europe and also in Thailand. And we're finding coronaviruses in these. And so lastly, I'm going to close with this. This was the story I was getting to. I was a slide ahead of myself because I glanced over to look at the clock. But have we missed some coronavirus outbreaks? And what should we be thinking about? And if you go back and you look in history, there was actually a cattle outbreak and widespread respiratory infection. The cattle had to be euthanized. There's widespread culling. It was a global pandemic for the cattle, but shortly after that, there was the Russian flu. And when you start to look at the symptoms of the Russian flu, you'll see some things that look very similar to coronaviruses. And the incubation period was much more in alignment with coronaviruses compared to influenza. And there's also the loss of smell. There was the protracted recovery. And if you start reading the case reports, you're going to see some things that actually will remind us quite a bit about COVID. So when you look at that proposal of whether or not it came from bovines, you think about OC43. And has OC43 really been in circulation for much longer? Was it really or was it really a relative to this Russian flu that we think might have been a coronavirus? Unknown, but it's food for thought. And it allows me to get here to the end of this, which is a little bit of my soapbox, which is we know that coronaviruses will continue to emerge. We know that the disease severity is likely driven by multiple factors, everything from the viral fitness, the receptor usage, to host factors. And we know that the risk of it becoming a pandemic is driven by multiple things. It's the transmissibility, the infectivity, the timing of the shedding. What is the background immunity in the population? Does it cross-react? Is there any? We know that coronaviruses will cause a wide range of disease. So we should expect to see a wide range of diseases from coronaviruses jumping into humans. And we know that, of course, that there are a lot more coronaviruses out there. So this is where I get a little bit on my soapbox, which is to say, if we understand differences between COVID-19 and SARS-CoV and MERS and our seasonal coronaviruses, as well as our veterinary coronaviruses or agricultural viruses, we'll have a much better chance of predicting the severity of the disease, as well as identifying treatments and development of countermeasures. So I will close there and say thank you to the speakers. I think I'm a minute over, so my apologies, but it's a pleasure to be here today and looking forward to questions. Thank you.